Alright, this is a fairly short lecture, but it involves a lot of memorization. You will know you're finished with Chapter 3 when you can describe the general structure and chirality of amino acids, name the 20 protein-related amino acids, their structure, three-letter code, and single-letter code, discuss the titration of amino acids, and describe the characteristics of the peptide bond. We live in a chiral world. Chirality, or handedness, is a really big deal in the natural world. Chirality refers to objects that are the same, but are mirror images to each other. Consider your hands for a moment. Your left and right hand are essentially the same, but you cannot superimpose one on top of the other. You can place them palm to palm, thus they are mirror images. Your hands are chiral. Amino acids, except for one, glycine, are also chiral. They possess a chiral carbon, and because they have four different substituents, groups connected to the carbon, they can be arranged to have mirror images. This matters because only one type of amino acid, the L enantiomers, are used in protein generation. The mirror images, D enantiomers, are degraded in the peroxisomes of cells and are not incorporated into proteins. A couple more examples to emphasize the point. Glyceraldehyde is a vital metabolite in intermediary metabolism. It's a precursor for glyceraldehyde 3-phosphate and is chiral, as is the amino acid alanine, as you can see here. Also note that to be chiral, a carbon must have four different substituents, not just four groups. For example, this is glycerol. The central point here is a carbon with four groups, three of which terminate in a hydroxyl and one not shown, that is, a hydrogen. However, this molecule is not chiral because two of the groups are the same, carbon hydroxide. Therefore, this molecule cannot be chiral, although it can become chiral just by changing one of those two groups. In addition to chirality, amino acids all have the same general structure. There is an amine group that's the NH3 and a carboxyl group that's the COOH. Remember your functional groups. These groups may or may not be protonated, as we'll see. Also, each amino acid has a side chain, or R group, that defines the characteristics of the amino acid. Glycine has only hydrogen, but it is still an R group in this case. On the next several slides, as well as in your book, are the canonical groupings and structures of the 20 amino acids, as well as their three-letter abbreviations and single-letter abbreviations. The 20 amino acids used in proteins can be classified as nonpolar or hydrophobic, polar, acidic or negatively charged, basic or positively charged. These figures are also found in your book, but I'm showing them here for completeness. The nonpolar amino acids are methionine, notice that sulfur methyl group on the end, leucine, tryptophan, proline, take good care to look at proline, phenylalanine, compare this structure to tyrosine, alanine, simple methyl, isoleucine, valine, glycine, that only has hydrogen as its side group. Next up are the polar uncharged amino acids. These are threonine, serine, cysteine, which is a bit unusual as you'll see. Notice the terminal sulfur. Asparagine, compare this one to aspartate. Tyrosine, look at this structure compared to phenylalanine. And glutamine, look at this compared to glutamate. While these are called acidic amino acids, typically under physiological conditions, they will have lost their proton and be referred to by their conjugate base names and carry a negative charge. Don't let this confuse you. They are considered the acidic amino acids. And those are aspartic acid, the conjugate base is aspartate, that's the typical name. Glutamic acid, its conjugate base is glutamate. There are three basic amino acids. Do note that at physiological pH these tend to be protonated and carrying a positive charge. We have histidine, lysine, arginine. Histidine is a bit odd, and its protonation state greatly depends on its local environment in a protein. However, under laboratory conditions at pH 7, histidine is 10 to 1 in its uncharged state. Also, as a side note, all amino acids, not just histidine, can have their properties greatly influenced by the protein environment. At this point, you should be working on describing the general nature of amino acids, including chirality and the basic concepts of their structure. Also, you should begin memorizing their structures, names, three-letter abbreviations, and single-letter abbreviations. Previously, when we studied neutralization or titration of simple weak acids, we saw a steady curve with a single inflection point. That single point is due to the existence of a single proton that was being examined. The acid deprotonated to its conjugate base, and the ratio of acid to conjugate base changed depending on increasing pH, as shown here. An amino acid is going to behave very differently from a simple weak acid, though in principle it is the same idea. Amino acids are weak acids, but they have multiple protonation sites. Consider the general amino acid structure. We're going to neglect the R group for a moment and focus on the amine and carboxy termini. At a low pH below 2, the amine and carboxyl are both protonated. This means the amine is NH3 and the carboxyl is COOH. The overall charge is +1. This fits with the idea that below their pKa, we can expect acids to be protonated. As the pH increases to 2.34, the first acidic proton on the carboxyl is lost, so we go from carboxylic acid to carboxylate. Notice the charge. The amine is still +1, and the carboxylate is now -1. So the overall charge of the molecule is 0. This is called a Zwitterion. It has two charges, but those charges cancel each other out. It was worth noting that the amino acids never have a protonated neutral state like a simple weak acid. They go from +1 to 0, and then, as we'll see, to -1. While there is a neutral state, it's a charged neutral state. Next, as the pH continues to increase, the amino will finally lose its proton. This occurs at around pH 9.7. Now the amine has lost its charge, and the carboxyl remains a carboxylate with a negative charge. The molecule is now charged at -1. You should also make a note of the pH values. At typical physiological pH, which is about 7 to 7.4, we should expect the amine and carboxy termini to be charged and the molecule to be a Zwitterion. This is useful to keep in mind when thinking about charges at physiological pH. Histidine is another unusual example. Again, we have the same amine and carboxyl ends that will deprotonate, but there is also the imidazole side chain. This idea will apply conceptually to any amino acid with a side chain that can accept or lose protons. The first acidic proton is, as expected, on the carboxyl group. This is lost at around pH 1.82, as shown, which brings the amino acid from a +2 net charge to a +1 net charge. Next, at about pH 6, the imidazole deprotonates, which removes our +1 charge, and we're now at a Zwitterion. Also the isoelectric point. Notice we have no net charge, but the molecule is charged by the amine and the carboxylate. So no net, but it's still a charged molecule. This will continue as we saw before, with a -1 charge appearing at pH 9.17. Why does that charge appear? I'll leave it to you to figure that out. Amino acids form long chains of peptides. The nomenclature is rather straightforward. A dipeptide is two amino acids connected by a peptide bond. A tripeptide is three amino acids connected by peptide bonds. An oligopeptide is a short chain of amino acids. It's a bit ambiguous. A peptide would be a longer chain of amino acids, and a protein is a gene product. Okay, so length is not really the question here, but whether or not the protein was formed from genetic instruction, that's a gene product, that is a protein, and may be one or more peptides involved in it. So how do amino acids form these chains? They use the peptide bond. The carboxyl group of one amino acid can bind to the amine group of the next amino acid by forming a water molecule as a leaving group. This creates a bond between the carbon and nitrogen. However, this carbon-nitrogen bond is not in and of itself a peptide bond. In other words, not just any carbon-nitrogen bond is a peptide bond. It requires additional characteristics. Remember, we're talking about a carboxylate here. So there is an associated oxygen. One oxygen is lost to the water molecule along with two hydrogens. The lone pair of electrons on the nitrogen can form a double bond with carbon, and this displaces those electrons on the carbon-oxygen system to become an additional pair of electrons on the oxygen. This gives us a Zwitter ionic style system and is also a resonance system. So the carbon-nitrogen bond is a partial double bond, as is the carbon-oxygen bond. This creates the rigid bond between carbon and nitrogen that is not anticipated by a simple carbon-nitrogen bond. In other words, for the peptide bond, we need the oxygen-carbon-nitrogen system. As a result, peptide bonds are typically planar and flat. So take a look at this generalized peptide chain. Notice a few things. First, there is an N-terminal residue. Residue is just another word for amino acid. Second, there is a C-terminal residue. These are also called the amino and carboxy termini, and peptides are always listed from N to C. There you have it. This brings us to the end of this lecture. At this point, you should be able to describe amino acid structures and chirality. You should be working on memorization of the names, structures, three-letter abbreviations, and single-letter abbreviations of the 20 common amino acids. We just discussed the titration of amino acids, especially in comparison to standard weak acids. And lastly, the peptide bond was discussed, as well as the general structures of peptides. For your practice and review, I suggest the following. Please do more than these in preparation for your work days. Remember, by the time you get to your work day, you should be ready to practice problems and seek help where needed. For review problems, I recommend all the questions in 3.1 and 3.2. For 3.3, I recommend 7, 8, 9, 15, 17, and 20. For 3.4, I recommend 24, 25, 26, 28, 29, 41, and 45. Good luck, and see you for the next lecture in Chapter 4.